Today's presentation will focus on our recent JITC paper showing that the kinase inhibitor dasatinib can reversibly switch off T-cell functionality following stimulation with T-cell bispecific antibodies. So this work was conducted at the Roche Innovation Center in Zurich under the supervision of Hélène Hagel and Christian Klein. So I would like to begin this presentation by giving a short introduction on the use of T-cell bispecific antibodies, which is a novel class of cancer immunotherapies. So as you can see on the illustration on the slide, T-cell bispecific antibodies can recruit T-cells to fight and eliminate the cancer cells. T-cell bispecific antibodies, or TCBs, are fully humanized IgG1 bispecific antibody with a 2-to-1 format. They are engineered with a bivalent binding to the tumor-associated antigens that allows for high avidity bindings to the target cells. In addition, these antibodies only have a monovalent binding to the CD3 epsilon chain of the T-cell receptor on T cells that avoids T-cell activation in the absence of any target cells. These antibodies also have an Fc reagent, which is engineered with a PG-LALA mutation that abrogates the Fc-related effector function while retaining the binding to the FcRn receptor for half-life extension. In this presentation, we will mainly focus on the HLA-2-WT1 TCB that targets WT1 peptide on AML tumor cells. On the illustration on the slides, you see that the WT1 TCB recognizes WT1 peptide presented on MHC class 1 by the AML cells. And at the same time, it can bind to the CD3 epsilon chain of the T-cell receptor on T cells, resulting in T-cell activation and the formation of a cytotoxic synapse where the release of cytotoxic granules can mediate the killing of the AML cells. However, the use of the WT1 TCB is also associated with an unpredictable risk of off-target off-tumor activity. Indeed, as I have shown you in the previous slides, the WT1 TCB can recognize WT1 peptide presented on MHC class 1 on the tumor cells, redirecting T-cell cytotoxicity towards the tumor cells. However, it could also be that the WT1 TCB recognizes peptide resembling WT1, which would be presented on MHC class 1 on healthy cells. In this case, the treatment with WT1 TCB would result in unwanted T-cell activation and tissue damage, which is called off-target off-tumor activity of the TCB. Therefore, in this presentation, we aim to verify whether the kinase inhibitor dasatinib would switch off T-cell cytotoxicity and cytokine release following treatment with WT1 TCB in the rare cases where the TCB would be active not on the target. So here we aim to verify whether dasatinib would interfere with downstream signaling pathways involved after TCR activation and whether this would result in a switch off of T-cell functionality, including T-cell cytotoxicity and cytokine release. So I will now move to the results of this presentation and highlight some of the experiments that are described in our JITC paper. So one of the first experiments that we conducted was an in vitro dose response of dasatinib in order to define the in vitro effective dose at which dasatinib is switching off tumor cell killing induced by WT1 TCB. Therefore, we conducted what we call an in vitro killing assay in which we co-cultured red fluorescent SKM1 tumor cells together with PBMCs, the WT1 TCB, and escalating concentration of dasatinib. We were able to follow the real-time killing of the red fluorescent SKM1 cells using an IncuCyte instrument. And we obtain the following killing curve shown on the right side of the screen, where we see in blue that in the absence of dasatinib, the stimulation with 10 nanomolar WT1 TCB resulted in tumor cell killing, and we then see that this tumor cell killing is abrogated with increasing concentration of dasatinib. We also see that for doses ranging between 50 to 100 nanomolar, dasatinib completely and significantly switched off the killing of the SKM1 cells by 10 nanomolar WT1 TCB. Therefore, the in vitro effective dose of dasatinib was found to be between 50 to 100 nanomolar dasatinib and could be translated into a clinical dose of 100 milligram given once daily to the patient, which would correspond to a Cmin of 2.6 nanogram per ml and a Cmax of 54.6 nanogram per ml. In another in vitro killing assay, we then investigated whether dasatinib would prevent T-cell activation following stimulation with WT1 TCB. Therefore, we measured the expression of CD25 and CD69 on CD4 and CD8 T cells by flow cytometry. And here you see that upon increasing concentration of dasatinib, the expression of CD25 and CD69 on both CD4 and CD8 T cells goes down. And it is completely switched off when the concentration of dasatinib reaches 25 nanomolar, indicating that dasatinib can prevent T-cell activation mediated by WT1 TCB stimulation. In the same assay, we also verified whether dasatinib would prevent WT1 TCB-mediated T-cell proliferation. Therefore, the PBMCs were stained with a CTV dye, and we could measure the dilution of the CTV dye in CD4 and CD8 T cells by flow cytometry as a readout for the effect on T-cell proliferation. So here, the histogram plots show the proliferation of CD4 and CD8 T cells following stimulation with 10 nanomolar WT1 TCB upon increasing concentration of dasatinib. So here you see for the CD4 T cells that the lowest concentration of 6.25 nanomolar dasatinib was sufficient to switch off the proliferation of CD4 T cells. For CD8 T cells, we see that we require the concentration of at least 25 nanomolar dasatinib to prevent proliferation of CD8 T cells, indicating that dasatinib had a stronger effect in reducing CD4 than CD8 T cell proliferation. In the same assay, we also assessed the effect of escalating concentration of dasatinib on cytokine release induced by WT1 TCB treatment, and therefore we collected the assay Supernatant and measured the cytokine levels by Luminex. So on this slide, you see the levels of TNF-alpha, IL-2, interferon gamma induced by 10 nanomolar WT1 TCB stimulation in the presence of escalating concentration of dasatinib. And here we see that when the concentration of dasatinib reaches 25 nanomolar, the release of TNF-alpha, IL-2, and interferon gamma was completely switched off, indicating that dasatinib was also able to switch off cytokine release following stimulation with WT1 TCB. In the second experiment, we wanted to mimic a clinical intervention with dasatinib and to verify whether dasatinib would stop further T-cell activation, proliferation, and cytokine release from pre-activated PBMCs. Therefore, we set up the following killing assay where we first activated PBMCs on SKM1 cells with WT1 TCB in the absence of dasatinib for 24 hours. At 24 hours, we added dasatinib in the system in order to verify whether this would prevent further release of cytokine and further T-cell activation in this assay. So in this slide, you see the effect of the addition of dasatinib on T-cell activation. So here in blue, you see the level of activation before the addition of dasatinib in the system, and in purple, you see the level of activation after the addition of dasatinib in the killing assay. In orange, you see the positive control where dasatinib was not added at 24 hours. So this graph shows that the addition of dasatinib prevented further expression of CD25 on CD8 and CD4 T cells and the expression of CD69 on CD8 and CD4 T cells. So overall, these data show that dasatinib was able to stop further T cell activation from pre-activated PBMCs. In the same assay, we then investigated whether dasatinib would stop further cytokine release from pre-activated PBMCs. So here you see in blue the levels of IL-2, interferon gamma, and TNF-alpha before the addition of dasatinib. In purple, you see the levels of IL-2, interferon gamma, and TNF-alpha after the addition of dasatinib. And here you see that both curves are overlapping, indicating that the addition of dasatinib could stop further release of IL-2, interferon gamma, and TNF-alpha in this system. In orange, you see that when dasatinib was not added in the system, we see higher release of IL-2, interferon gamma, and TNF-alpha, which was happening between 24 and 48 hours in the killing assay. This experiment was also repeated for additional donor, and here we saw the same trend that the addition of dasatinib in the system prevented further release of IL-2, interferon gamma, and TNF-alpha. Last, we assessed the effect of dasatinib on T cell proliferation in this assay, and therefore we measured the dilution of the CTV dye as a readout for the effect on T cell proliferation by flow cytometry. So the histogram plot showed the effect on CD4 and CD8 T cell proliferation. So in black, you see the negative control in the absence of WT1 TCB, and in blue, you see the positive control where dasatinib was not added in the system following stimulation with 10 nanomolar WT1 TCB that induced proliferation of CD4 and CD8 T cells. In pink, you see the conditions where dasatinib was added after 24 hours of activation in the system, and you see here that dasatinib delayed the proliferation of CD4 and CD8 T cells. We also measured the count of CD8 and CD4 T cell at the assay endpoint in order to verify whether dasatinib prevented proliferation of CD4 and CD8 T cells. And here in purple, you see the conditions where dasatinib was added in the system. And in this condition, you see that the count of CD8 and CD4 T cells was much lower than in the absence of dasatinib, indicating that dasatinib was efficiently preventing further T cell proliferation from pre-activated PBMCs. We then wanted to investigate how dasatinib was switching off the killing of the target cells mediated by WT1 TCB stimulation. And therefore, we measured the T cell degranulation marker CD107A on CD4 and CD8 T cells by flow cytometry following stimulation with 10 nanomolar WT1 TCB in the presence and in the absence of dasatinib. So here on the flow cytometry plots, you see the expression of CD107A on CD4 and CD8 T cells after stimulation with WT1 TCB in the absence of dasatinib. So here we see that we have positive cells for CD107A among CD4 and CD8 T cells. However, when we added dasatinib together with WT1 TCB, we see that the proportion of CD107A positive cells among CD4 and CD8 T cells is strongly reduced. This is also highlighted by the bar plot shown on the right side of the screen, where you see the data for 3 donors. And in pink, you see that when dasatinib was added in the system, the expression of CD107A on CD4 and CD8 T cells was strongly reduced, indicating that dasatinib was able to stop T cell degranulation, stopping the release of perforin and granzyme B, which are required for the killing of the target cells after stimulation with WT1 TCB. In the third experiment, we aim to verify whether dasatinib could stop further tumor cell killing from pre-activated PBMCs, which was not shown in the previous experiment since all of the target cells were killed after 24 hours of activation. So therefore, we set up a restimulation assay in which we first pre-activated PBMCs on CFSC-labeled SKM1 cells in the presence of WT1-TCB and in the absence of dasatinib. We then collected these activated PBMCs and stimulated them on fresh CTV-labeled SKM1 cells with WT1-TCB, this time in the presence of dasatinib, to verify whether dasatinib would stop further tumor cell killing from these pre-activated PBMCs. So as a readout for this assay, we measured the killing of the CFSC-labeled SKM1 cells used upon the first stimulation in the absence of dasatinib, and the killing of the CTV-labeled SKM1 cells used upon the second stimulation in the presence of dasatinib. And the killing of these CFSC and CTV-labeled SKM1 cells was measured by flow cytometry using a live dead stain. So here you see on the flow cytometry plots the percentage of dead and live cells following stimulation with WT1 TCB upon the first and second stimulation. So in green, you see that most of the CFSC labeled SKM1 cells were efficiently killed by WT1 TCB upon the first stimulation in the absence of dasatinib. However, upon the second stimulation, you see that the CTV labeled SKM1 cells were not killed in the presence of dasatinib, indicating that dasatinib could stop further killing of target cells by pre-activated PBMC. On the right side of the screen, you also see the dose response curve showing the killing of the SKM1 cells over the concentration of WT1 TCB. And here in green, you see that all of the CFSC labeled SKM1 cells used upon the first stimulation in the absence of dasatinib were efficiently killed, as upon the second stimulation, none of the CTV labeled SKM1 cells were killed in the presence of dasatinib, here shown in pink. In purple, you see the positive control where dasatinib was not added upon the second stimulation. In this assay, we also collected the supernatant after the second stimulation in the condition in the presence and in the absence of dasatinib, corresponding to the on-off switch. So here you see that when dasatinib was added in the system, the off condition, it completely prevented further release of IL-2, interferon gamma, TNF-alpha, IL-8, GM-CSF, and IL-6. So these data show that dasatinib was able to stop further cytokine release by PBMC that were pre-activated with WT1 TCB. So overall, this experiment was the proof of concept that dasatinib could be used to mitigate off-target, off-tumor activity of WT1 TCB. But in the last in vitro experiment, we wanted to verify whether the inhibitory effect of dasatinib were reversible and whether the removal of dasatinib would result in tumor cell killing. Therefore, we set up a restimulation assay with 3 different stimulation steps using CA-TCB and MKN45 tumor cells and PBMCs, and we try to mimic off-on-off switch by either adding dasatinib upon the first stimulation, then removing dasatinib upon the second stimulation, and adding dasatinib again upon the third stimulation. We also mimicked an on-off-on switch by not adding dasatinib upon the first stimulation, then adding dasatinib upon the second stimulation, and removing dasatinib upon the third stimulation. And as a result, we obtain the following killing curve that were measured by IncuCyte, where we see the off-on-off switch, where the addition of dasatinib upon the first stimulation prevented the killing of the MKN45 tumor cells. When dasatinib was removed upon the second stimulation, the killing of the MKN45 cells was restored, and when dasatinib was added upon the third stimulation, the killing of the target cells was switched off, indicating that the effect of dasatinib were reversible and that dasatinib reversibly switched off CA-TCB-induced tumor cell killing. we saw the opposite effect in the on-off-on switch condition. In this assay, we also measured the cytokine levels in the supernatant by Luminex after the first and second stimulation in the presence and absence of dasatinib. So here you see the levels of cytokine in the off-on switch. So when dasatinib was added upon the first stimulation, there was no release of IL-2, interferon gamma, and TNF-alpha. However, when dasatinib was removed upon the second stimulation, the release of IL-2, interferon gamma, and TNF-alpha was restored. We could see the opposite effect in the on-off condition. So these data indicate that dasatinib was able to reversibly switch off cytokine release induced by CA-TCB treatments. So I will now move to the work that was conducted using humanized NSG mice in order to show that dasatinib would prevent B-cell depletion and cytokine release following treatment with CD19-TCB. So here we aim to verify whether the short PK/PD properties of dasatinib in combination with the long PK/PD properties of CD19-TCB would efficiently prevent cytokine release and activity of the TCB in vivo. So therefore, we used humanized NSG mice, non-tumor bearing humanized NSG mice, and treated them either with vehicle, CD19-TCB given as a monotherapy, or CD19-TCB given in combination with dasatinib that was given 2 or 3 times per day to the mice. At 48 hours and 72 hours, we measured the CD20 plus B cell count in the peripheral blood of the mice as a readout for the effect on efficacy. We also collected blood at earlier time point to collect serum and measure cytokine release induced by CD19-TCB treatment. So in this slide, you see the count of CD20 plus B cells in the peripheral blood of the mice, 48 hours and 72 hours post-treatment with CD19-TCB. In blue, you see that CD19-TCB is a monotherapy induced a complete depletion of B cells already 48 hours after the first treatment with CD19-TCB. In purple, you see that when the mice were also co-treated with dasatinib, none of the CD20 plus B cells in the peripheral blood were depleted, indicating that dasatinib completely switched off the activity of CD19-TCB. At 72 hours, we saw a partial depletion of the CD20 plus B cells in the group of mice that was co-treated with CD19-TCB and dasatinib. And here we believe that the exposure of dasatinib was insufficient at 72 hours to completely prevent the activity of CD19-TCB. And since the effects of dasatinib are reversible, we believe that CD19-TCB was able to partially deplete some of the B cells. We then investigated the effect of dasatinib on cytokine release in these humanized NSG mice following treatment with CD19-TCB. And therefore, we measured the cytokine levels in the serum of the mice by Luminex. So here you see the levels of IL-2, TNF-alpha, interferon gamma, and IL-6, 1.5 hours and 6 hours post-treatment with CD19-TCB. In purple, you see that in the group that was co-treated with dasatinib, there was no release of IL-2, TNF-alpha, interferon gamma, and IL-6 at both time points, indicating that dasatinib completely switched off cytokine release induced by CD19-TCB in humanized NSG mice. In the second in vivo experiment, we then wanted to verify how dasatinib compares to glucocorticoid, JAK, and mTOR inhibitor, which are mitigation strategies used to prevent CRS. So here we used non-tumor bearing humanized NSG mice that were either treated with vehicle, CD19-TCB as a monotherapy, or CD19-TCB in combination with the JAK inhibitor ruxolitinib, the SRC inhibitor dasatinib, the mTOR inhibitor sirolimus, or the glucocorticoids methylprednisolone or dexamethasone, which are currently used in the clinic as mitigation strategies against CRS. So here the JAK and mTOR, the JAK and SRC inhibitor were given twice on day 0 and then twice on day 1 and day 2. As methylprednisolone, dexamethasone, or the mTOR inhibitor were given twice on day 1 and then twice on day 0 and then once on day 1 and once on day 2. We then collected the serum of the mice 6 hours post-treatment with CD19 TCB to measure the effect of the different treatment on CD19 TCB-induced cytokine release. So here on this slide, you see the effect on interferon gamma, IL-2, TNF-alpha, and IL-6 release. And what we can see is that most of the mitigation strategies strongly reduce the levels of these 4 cytokines, but dasatinib appears to be the most potent mitigation strategies that reduces interferon gamma, IL-2, TNF-alpha, and IL-6 release. We then measured the count of CD20 plus B cells in the peripheral blood by flow cytometry as a readout for the effect on efficacy. And here we see that most of the mitigation strategy minimally interfered with B cell depletion induced by CD19 TCB. We see that the effect was a little bit more pronounced for the group that were co-treated with dexamethasone, sirolimus, ruxolitinib, and dasatinib. However, the effect, the inhibitory effect of dasatinib were not much stronger than what we observed in the group co-treated with dexamethasone, sirolimus, and ruxolitinib, indicating that dasatinib might not interfere so much with anti-tumor efficacy. So in the last in vivo experiment, we then wanted to assess the effect of dasatinib on long-term anti-tumor efficacy, and therefore we used a lymphoma patient-derived xenograft in humanized NSG mice. We also compared the effect of dasatinib to glucocorticoid, ruxolitinib, and sirolimus in this model. So here, the humanized NSG mice were engrafted with a lymphoma PDX, and once the tumors were big enough, we then treated the mice with vehicle CD19 TCB as a monotherapy or CD19 TCB in combination with sirolimus, dexamethasone, ruxolitinib, or dasatinib. So here the mice received 1 injection of CD19 TCB per week, and upon the first injection with CD19 TCB, which is associated with the highest risk of cytokine release, we also co-treated the mice with the different mitigation strategies. So here, dexamethasone was given twice on day 16 and then once on day 17 and day 18. The same for sirolimus, and for ruxolitinib and dasatinib, which are short-lived molecule, we gave them twice on day 16, twice on day 17, and twice on day 18. As a result of this experiment, we obtained the following tumor growth curve. So here we see in gray in the vehicle group that the PDX were nicely growing in the mice, and we see in blue that CD19 TCB as a monotherapy induced a nice anti-tumor activity. We see in red that in the group that was co-treated with dasatinib, dasatinib minimally interfered with anti-tumor efficacy, and we also see that the effect of dasatinib was comparable to the other mitigation strategies, consisting in ruxolitinib, shown in purple, sirolimus, shown in light blue, and dexamethasone, shown in brown. So overall, the transient use of dasatinib upon the first infusion with CD19 TCB did not completely prevent anti-tumor efficacy mediated by CD19 TCB treatment. On the right side of the screen, you also see the individual tumor growth curve for each of the mice in each of the group, just to highlight that there is a lot of variability in this model, but that overall CD19 TCB was nicely controlling and inhibiting tumor growth. We also see that in the group of mice that were co-treated with dasatinib, the dasatinib slightly interfered with anti-tumor efficacy mediated by CD19 TCB in comparison to the other mitigation strategy consisting in ruxolitinib, sirolimus, and dexamethasone. So I now come to the last slides of this presentation, and I would like to summarize the key data shown in the presentation. So one of the important findings of this presentation is that dasatinib can reversibly switch off T-cell functionality following stimulation with T-cell bispecific antibodies. Therefore, in an in vivo setup, when the exposure of dasatinib is not sufficient anymore, the TCB can take over and induce T-cell cytotoxicity. So as take-home messages for this presentation, it was shown that dasatinib is reversibly effective at the pharmacological active dose of 100 milligram given once daily. That dasatinib can efficiently stop T-cell proliferation, T-cell activation, and cytokine release following stimulation with WT1 TCB. But in addition, it was also shown that dasatinib can also prevent further target cell killing and cytokine release by pre-activated PBMCs, mimicking a clinical intervention with dasatinib. Last, we have shown that dasatinib efficiently prevents B-cell depletion and cytokine release in humanized NSG mice that were treated with CD19 TCB. So overall, this data suggests that dasatinib can be used as a mitigation strategy for the unpredictable risk of tumor activity, which is associated to the treatment with WT1 TCB, or that it could be used against high-grade CRS associated with T-cell engaging therapies in general, where we would want to fully switch off the activity of T-cells. Additionally, we have shown that the transient use of the SRC inhibitor, mTOR inhibitor, and JAK inhibitor could represent an attractive approach to mitigate TCB-induced cytokine release while retaining the efficacy. With that, I would like to thank you all for joining this presentation, and also thank all my colleagues from Roche who contributed to this project, and my academic supervisor from the University of Basel.